Hello, my name is Chemistry, your research helper. I know you are saying enough of this introduction, just come to the point, so let's start. The paper we are going to understand today is a research article from the journal Electrochemistry, focusing on the electrode reactions of copper species in an ionic liquid called 1-butyl-1-methylpyrrolidinium bisfluorosulfanilamide BMPFSA. The authors, Nobuyuki Serizawa, Takumi Hisada, and Yasushi Katayama from Keio University, have conducted experiments to understand the electrochemical behavior of copper in this specific ionic liquid, which has potential applications in redox flow batteries. Based on the provided document, the experiments conducted involve cyclic voltammetry, UV-vis spectroscopy, and the electrodeposition of copper in the ionic liquid BMPFSA. Here is a detailed step-by-step procedure for each experiment. Experiment 1. Cyclic voltammetry of copper in BMPFSA materials needed. Copper electrode 1-butyl-1-methylpyrrolidinium bisfluorosulfanilamide BMPFSA AGAGI reference electrode potentiostat electrochemical cell glassy carbon GC and platinum PT electrodes procedure preparation of electrochemical cell. Clean the copper electrode thoroughly. Assemble the electrochemical cell with the copper electrode as the working electrode, AGAGI as the reference electrode, and a suitable counter electrode. Solution preparation. Prepare a solution of BMPFSA containing 50 ml dm cubed CUI. Cyclic voltammetry. Set up the potentiostat and connect it to the electrochemical cell. Perform cyclic voltammetry at different scan rates with IR compensation. Record the current response to the applied potential. Data analysis. Analyze the cyclic voltammograms to identify oxidation and reduction peaks. Note the potential values where copper dissolution and deposition occur. Experiment 2. UV-vis spectroscopy of BMPFSA with copper materials needed. UV-vis spectrophotometer cuvettes BMPFSA solution with varying concentrations of CUI procedure, sample preparation, prepare BMPFSA solutions with different concentrations of CUI. Spectroscopy, fill the cuvette with the sample solution. Measure the UV-vis spectrum over the specified wavelength range to 0.0500 nanometer. Data analysis, analyze the spectra to observe changes in absorbance with varying copper concentrations. General safety and cleanup. Follow all safety protocols, including wearing appropriate personal protective equipment. Dispose of chemical waste according to your institution's guidelines. Clean all equipment thoroughly after use. Concentrations. The concentration of CUI in BMPFSA for cyclic voltammetry is 50 ml dm cubed. For UV-vis spectroscopy, prepare BMPFSA solutions with varying concentrations of CUI to observe the absorbance changes. This guide provides a comprehensive approach to replicating the experiments described in the article. Thank you.